your renal. Okay, in general, is to maintain homeostasis. That's it. In the ECF. Okay, maintain. But then we go deeper. Okay, how it maintains the homeostasis. So first we have the regulations of blood pressure. How? By regulating this ECF volume. And to save important substance, okay, the blood that goes through the kidney, all the substance will be filtered and then reabsorbed back to the body, okay, such as the glucose, the amino acid, and bicarbonate. And at the same time, also the filtered, the non-functional substance, so it will be excreted. Okay, examples like toxic substance, the waste products, some of the drugs, okay, from the body by filtration, secretion, and then at the end excreted out of the body in form of the urine. So in the urine contain all these toxic and waste products. And at the same time also it functions to secrete the hormone, okay, secretion of hormone, Renal, which is for the blood pressure regulation. We have erythropoietin for the RBC productions, okay, vitamin D for the absorptions of calcium ion, then prostaglandins, infrarenal regulations of this renal blood flow. And the kidneys also functions to maintain this acid-base balance in the body. So you will have a specific lecture later about this acid-base. And then to maintain the osmolarity of this ECF, and to regulate the water and electrolytes balance in the body. So all of this is the specific functions of the kidney. So how is the urine being formed? Okay, formation of the urine. So we have steps here. Okay, we start with the filtration. Okay, filtration is the first step in the urine formation. What is meant by filtrations? The blood that passes through the nephrons, the glomerular capillaries later will be filtered. The substance will be filtered into the lumen, and then some of the substance will be reabsorbed. We have reabsorptions. Okay, the movement of these compounds from the filtrate across the tubule epithelium back into the peritubular capillaries. So that's what we call reabsorptions. So at the same time, we also have a secreted. So the movement is from the peritubular capillaries into the lumen of the tubules. Okay, so jangan terbalik. So reabsorptions from the lumen to the capillary, secretions from capillary to the lumen. Okay, and then the glomerular filtrate after is modified, so there are a few modifications between reabsorbs and secretions. Finally, it will be, the final product will be excreted, okay, from the body as a urine. So this is the basic steps of the formations of urine. So we back to the anatomy, just to see from the gross. Okay, two separate kidneys. We have two separate kidneys, okay, located at the upper abdomen, sorry. And then, okay, first you have renal artery that supply kidneys with the blood, and then the renal vein that carry the blood filtered by the kidneys, and go down to the ureters that carry urine to the urinary bladder. And then it will stop the urine temporarily in the urinary bladder, and finally excreted through the urethra. From the cross, we go deeper to the parts of the kidney. So just to refresh back, we have three distinct regions: the outermost renal cortex, the middle renal medulla, and inner renal pelvis. Okay, so you need to refresh back, because later once we look at the nephrons, the type of the nephrons, you will know we have two types of nephrons at the different region. Okay, so what are the the nephrons. So in physiology, we look more on the nephrons. There are more than 1 million of nephrons in the kidney. So to carry out, what's the functions? Is to carry out the functions of the kidney. The nephrons together with the renal circulatory system will provide all the process that I mentioned just now, the filtration, the reabsorptions, and secondary process. So the nephrons you can say as the functional units of the kidney. So from this, we take out one nephron. So we have millions of nephrons. Okay, so this is one nephron, okay, basic structure. So each nephron has two components, vascular and tubular. Okay, these two works with each other, vascular and tubular. And if we look at the arrangement of nephrons within kidney, it will give rises to two distinct regions. Okay, we divided into outer regions, the renal cortex, and the inner regions, the renal medulla. Okay, now we zoom in lagi, we look at the vascular component. Okay, I need you to familiar with this vascular and tubular because when we go deeper, we will see a different mechanism for each vessels and each tubule. Okay, so get it into your mind, your memory, and save. Alright, so vascular component, we start with the afferent arteriole. Okay, we start with the afferent arteriole that carries blood to the glomerulus. Okay, it starts from there. Okay, it carries blood to the glomerulus. Okay, and then this glomerulus, you nampak macam bola-bola benang macam tu, capillaries, a tuft of capillaries. Okay, the functions is to filter. Okay, this is the filter here. Okay, filter a protein-free plasma into the tubular component. Okay, that this brown color is all this tubular. Alright, and then you have the efferent. Okay, the name E means they carry blood from glomerulus. So the acid, okay, so carry blood from glomerulus. And then you see all this web of capillaries, these peritubular capillaries, okay, is to supply the renal tissues involved in exchange with the fluid in the tubular lumen. So there are processes here, okay, the exchange along these capillaries and also the tubule that it passed through. Good, so that is a vascular component. Now we look at the tubular component. First is the Bowman's capsule. So here, macam U-shape ni. Okay, Bowman's capsule. It collects the glomerular filtrate. Okay, tengok kedudukan. So dekat sini ada glomerulus tadi. So it filter all the filtrates who goes into the Bowman's capsule. From there, it goes to the proximal tubule. Okay, where it uncontrolled reabsorptions and secretions of selected substance occur here. And then you have to go past through the loop of Henle. Okay, this U-shape loop of Henle, establish osmotic gradient in the renal medulla that is important in the kidney's ability to produce urine of varying concentration. So we're gonna look in specific here. In the next lecture, we're going to look on the countercurrent mechanism along this loop of Henle, how it dilutes and concentrates the urine. Okay, and then you have the distal tubule and the collecting duct. Okay, so this distal tubule and collecting duct is variable. It controls the reabsorptions of sodium and water, secretions of potassium and hydrogen ion occurs here. And then the fluid leaves the collecting duct is the urine, which enters the renal pelvis. Okay, in here we also have a special structure, the juxtaglomerular apparatus. Okay, kat sini point dia bertembung. Okay, it's connected here. Produce substance involved in the control of kidney function. So don't worry, we'll go detail. Okay, let's see if we can play around these things. Cannot play, so remove. Sorry. Okay. All right. Okay. Back to the circulatory system of the kidney. Again and again. Okay. Approximately 20 to 25% of cardiac output will goes to the renal artery. Goes into the nephrons. Okay. Again, it goes to it will pass the afferent arteriole, and then filtered glomerular capillaries. And then as it afferent arterioles, and then you have the peritubular capillary that surround tubular part of the nephrons. And the filtrate will go pass through the Bowman's capsule. Okay. So if we go back, okay, do you notice the arrangement? So first we have two sets of arterioles and two sets of capillary. So if you go look at the capillaries, is the glomerulus and the peritubular capillaries. They have a different function. The glomerulus, again and again, to filter, okay, for filtration. And then the peritubular capillaries is for reabsorption and secretion, specifically. And in terms of the arterioles, just now we have afferent and efferents. They provide the resistance. Okay, they are the resistant vessels. Okay, when you look at the control of blood pressure, later you're gonna look how it provides the resistance. So afferents is to regulate the amount of blood flowing through the glomerulus. Okay, remember the structures, then you will know the functions. Okay, and then the efferent arterioles provide resistance and regulate the blood flowing out of the glomerulus. So the constriction, okay, if the constrictions of the efferent here, we constrict, okay, will cause more blood to accumulate in the glomerulus, more time for filtration to occur in this glomerulus. So there is the creating we call as hydrostatic pressure that enhance filtration. So we're gonna look this in the Starling forces about the filtration in the GFR. Okay, so this is the important arrangements for the renal function. Now, this is what I mentioned. We have two types of nephrons. Okay, distinguished by location and the length. Okay, where it's located and how long it is. Okay, we have a cortical nephrons and juxtamedullary nephrons. Okay, by the picture itself, just illustrations. So if you look at the cortical nephrons here, pendek, the short. Okay, the most abundant type of the nephrons. And if you look at this figure, this side, this is juxtamedullary nephrons. You can see it's a very long loop nephrons. Okay, this juxtamedullary nephrons is important to establish the medullary vertical osmotic gradients. Okay, you see the location. Okay, juxta will go deeper into the medulla and a bit longer. And they have a different function. So what are different functions? So again, cortical nephrons yang short tadi, okay, pendek je sini, untuk for the filtration, reabsorption, secretion. Okay, it's the majority of the nephrons is the cortical nephrons. So how about juxtamedullary nephrons? Why we have these longer nephrons? Okay, for the dilutions and concentrations of the urines. Okay, so how the mechanism is through maintaining the hypotonic medullary institution. Okay, so there are two, okay, cortical and juxtamedullary. So again, back to the basic, again and again, we have glomerular filtration, tubular reabsorption, and tubular secretion. So I don't have to repeat. You should be able to know now the movement from where to where. Okay, and so don't get it mixed up. If you mix up, you get wrong for everything of the concept. Okay, so we go in details. First, the glomerular filtration. Okay, it happens in the name itself, in the glomerulus. Okay, the capillaries just now in bola ni. Okay, glomerulus. Inside where? In the Bowman's capsule. Okay, that's the location. Okay, there is, kalau kita zoom kat sini apa yang ada, the structures, there is a hole. Okay, dia macam ada lubang, filtration kan, bayangkan filter. So there is a hole in the glomerular capillaries, what we call as a fenestrate. So they make capillaries more porous than other capillaries in the body. So it has podocytes to allow everything except blood cells dengan protein to live. Okay, that's why in the urine you never see blood right inside there, because it cannot pass this filter. Okay, and protein also cannot pass through. So the rest is the solutes. So that's why it has this podocyte. And then the liquid that has left the blood supply here is called the filtrate. Okay, that's the term, filtrate, and essentially the same as interstitial fluid in the composition. Okay, flow and filtration rate. Okay, just now mentioned about the hydrostatic pressure. So what pressure we have inside this? We have hydrostatic and oncotic pressure, or osmotic, oncotic or osmotic. Okay, which will determine the filtration across the glomerular capillary. Okay, glomerular capillary filtration depends on the balance of this hydrostatic and oncotic. Okay, hydrostatic pressure at the afferent end of the glomerular capillaries is high and decrease. Okay, we start. Alright, hydrostatic pressure of the glomerular capillaries start with the high and then it will decrease. Okay, slightly along the length of glomerular capillary. For the oncotic pressure, the plasma oncotic pressure in glomerular capillary increase along the length of the glomerular capillary as plasma is filtered. So at the large protein that remains, remember protein cannot pass through. So once they pass the filtration, protein semakin bertambah. So bila makin tinggi protein, dia akan increase the plasma oncotic pressure. So the net balance of pressures will ensure only filtration occurs in the glomerular capillaries. So between these hydrostatic and oncotic. We will look later in details in the GFR. Okay, if you look at the structures, the afferent arteriole is larger than efferent arterioles. Why we need afferents bigger? Because the blood supply leaving the glomerulus will have part of the volume removed due to the filtering. So we need to keep pressure in the glomerulus high enough that the filtrate can leave the glomerulus efficiently. So you cannot have the same resistance. If afferent is same diameter with the efferents, so how you want to give a high pressure for the filtrate to goes to the Bowman's capsule? So you need one bigger from afferent to enter, and efferent a bit like small, so that it will give longer time and increase the pressure inside, so that the filtration will be efficient. Okay, again, the blood flow to the kidneys is about 20 to 25% of cardiac output. Okay, normally the amount of pressure inside the glomerulus is 55 milligram mercury. Okay, so here glomerular blood hydrostatic pressure normally is 55. Okay, there are counter pressure. Dia mesti masuk misalnya pressure yang counter dia. Okay, where the water tries to go back, okay, into the glomerulus. Okay, the pressure is try to go back into the glomerulus because of the osmosis. So this is what we call the colloid osmotic pressure. Okay, around 30 milligram mercury. So dia mesti ada oppose, dia nak masuk balik. Okay, there is also a capsular pressure. that comes from the walls of the Bowman's capsule, resisting overfilling by the filtrate is around 50, isn't it? Okay, and then the net pressure. So overall, we have outward pressure sending the filtrate down the proximal tubule around 10. So this is how we calculate the net filtration pressure. Okay. So if the pressure get out of balance, then renal shutdowns is very possible because blood cannot be cleansed. Or it can be the pressure drop due to hemorrhage or severe dehydration. So we need to have the net filtration pressure here normally. So that's about the filtration. Next step, we move to reabsorption. Okay, tubular reabsorption. Okay, the filtrate is very similar to blood plasma without the proteins. Okay, remember we don't have any proteins filter. Okay, at this point, not all like urine. Okay, if this filtrate excreted straight away, body will lose more than 10 times. So you need to reabsorb because in filter, when it filter, it doesn't select it. You just filter as long as you can pass through the pores and as long as you fulfill the criteria of this glomerular filtration. Okay, it's not selective. So when it passes through as a filtrate, the body need to reabsorb back because you don't want to lose the solids that are important for the body. Okay, so tubular reabsorption mechanisms in the nephrons will return the water and solids that the body needs back into the extracellular fluid and the circulatory system. So that's the functions of the tubular secretion. Tubular reabsorption, sorry. Okay, in the step 3, we look at the tubular secretion. Okay, tubular secretion from the blood into the tubular fluid. Okay, so nephrons are also able to secrete unwanted substance from the bloodstream into the filtrate. Okay, so together this process complete the transformations of the glomerular filtrate into the urine. So along the tubule and the blood capillaries in the structures, there is a reabsorption, secretions, and they try to balance each other. And then at the end, the waste will go out through the excretion. Okay, this is just go details again about the pores, okay, about the filtration, the characteristic of this glomerular filtration. So again, we know the fluid filtered from the glomerulus into the Bowman capsule will pass through 3 layers of this glomerular membrane. Okay, there are 3 layers. First, we have the glomerular capillary wall, which is more permeable to water and solutes than capillaries elsewhere in the body. It's highly fenestrated. It stops cellular component of blood coming into contact with the basement membrane. And then we have the basement membrane. It's composed of collagen and glycoproteins, and it is negatively charged. Okay, and then we have the inner layer of Bowman's capsule, consists of podocytes that encircle glomerular stuff to act to maintain basement membrane. So in here, so you can see the clue here. The main criteria is size and also the charge. Okay. So here we know that just now from the structures, the layers, okay, the filtration affected by the charge and molecular weight. Okay, in terms of the size, okay, the molecular weight. Usually the diameter of 8 nanometer will not filtered. Okay, these some books say 7, 8, but it doesn't matter. So as long as you know the size, is the diameter, the bigger diameter is not filtered. Okay, and then the molecular weight. Okay, examples like albumin. Albumin diameter is 6 nanometer. By Rex, it should be passed through. Okay, but the charge, okay, the charge doesn't allow to be filtered. Okay, negative will not filter easily. Okay, So albumin dextrans have negative charge. So again, dia tak boleh fit the criteria, so it won't be filtered. Okay, the albumin. So what happens if in the case of proteinuria or albuminuria, where you found the albumin or protein in the urine? So why? Because the negative charge of the basement membrane are lost. Okay, there's some impact in terms of the charge, okay, in the membrane. So result some of the lower molecular weight proteins, especially albumins, but they will be small tadi. And then, but then this is the impact in terms of the membrane just now, the negative membrane impact. So they boleh filtered, it pass through, and then its presence in the urine. Alright, don't confuse this juxtaglomerular apparatus with juxtaglomerular nephrons. Okay, it's different things. So juxtaglomerular apparatus (JGA) is to maintain blood pressure and act as quality control mechanism to ensure proper glomerular filtration rate and efficient sodium reabsorption. Okay, where is it? This here at this blue color. Okay, so this juxtaglomerular apparatus consists of the afferent arteriole, efferent arteriole, and distal tubule associated with that glomerulus. So if the delivery of the sodium chloride to the distal tubule fails, the afferent arteriole will dilate and renin, okay, and renin will release, causing formation of the angiotensin 2. And at the same time, it will constrict the efferent arteriole. So these changes will cause increase in the glomerular filtration rate and increase the sodium chloride load to restore back the sodium chloride delivery to the macula densa. Okay, so that's how it works for the JGA in terms of the sodium chloride. Okay, so it's sensitive to the sodium chloride concentration that we filtered. So now we look on the control of the renal functions. Okay, in terms of the circulation, the lymphatic innervation, the blood flow, and also the plasma flow. So again, so kidney receive 20 to 25% of total cardiac output. So these fractions, okay, this 20 to 25% is known as the renal fraction. Okay. So again, what does it mean by renal fraction? Is the part of this cardiac output that passes through the kidney. Okay. In average adults, okay, with the cardiac output of 6 liters per minute, the RBF is around 1.2 liters per minute. Okay, it depends how many percent you take there, 20 to 25%. So again, just to again and again to remind you back, we have afferent, efferent arterioles and glomerular capillaries, and either peritubular capillaries or vasa recta. Okay, peritubular capillaries is the one that surrounded the cortical nephrons. The vasa recta is the one that surrounded the juxtamedullary nephrons. So vasa recta of juxtamedullary nephrons is specifically vasa recta is present in the juxtamedullary nephrons loop, okay, to give the kidney's ability to produce a concentrated urine. What's the functions? To deliver blood to medullary cells, return the reabsorbed solutes and water in the medulla to the general circulation without disrupting the medullary concentration gradient. So it has a specific function, okay, different from the peritubular capillaries. Okay, this is again repetition. So we have the afferent arterioles that regulate the amounts of blood entering glomerulus by controlling its diameter. Okay, and then you have the efferent arteriole that is smaller diameter than afferents because it needs to give the resistance to create the hydrostatic pressure and controls the blood exiting the glomerulus. And 20 to 25% of plasma entering kidneys were converted, or 20% of plasma entering kidneys converted to glomerular filtrate. I don't have to go through this. This one is just back to the anatomy, to the renal circulation, where you start again from the afferent to the end. So what we want to highlight here is the important features in organizations of renal microcirculation, where you have the glomerular capillary bed supplied by the afferent arteriole and drained to the efferent arteriole. You have the peritubular capillary bed that receive reabsorbed water and solutes, and long peritubular capillary loops that known as vasa recta involved in the urine concentrating mechanism. Okay, again, if you back to the concept, why we have these short renal arteries, okay, and then presence of afferent arterioles, again, is to provide the hydrostatic pressure in the glomerular capillaries that helps in the filtration process. Okay, renal innervation by renal plexus. It starts branch of the celiac plexus. It has sympathetic fibers and control the diameter of the renal arteries, hence urine forming functions. Okay, increase in the renal sympathetic nerve activity will triggers the vasoconstrictions in kidney, which decrease the renal blood flow and decrease the urine sodium excretion, so does increase the renal secretions. In terms of the drainage, okay, is a renal lymph vessel that starts in renal cortex, parallel to arcade veins, leave the hilus, brains, thoracic duct into venous circulation, and it functions to return protein reabsorbed from tubular fluid to the duct. So this is just good to know, okay, the circulation and things. That's all about your kidney. Boleh? Dah boleh dah? Okay? So dah boleh imagine the